James Dewey Watson was born in Chicago, Illinois on April 6, 1828, as the only son of James D. Watson and Jean Mitchell. Watson's entire boyhood was spent in Chicago, where he attended Horace Mann Grammar School for 8 years and South Shore High School for 2 years. He then received a tuition scholarship to the University of Chicago, and in the summer of 1943 entered their experimental 4-year college. In 1947, he received a bachelor's degree in zoology. During these years, his boyhood interest in birdwatching had matured into a serious desire to learn genetics. At Indiana University, he received his Ph.D. degree in zoology in 1950. Here, Watson studied the effect of hard X-rays on bacteriophage multiplication. From September of 1950 to 1951, he spent his first postdoctoral year in Copenhagen as a Merck Fellow of the National Research Council. At the zoological station, he saw for the first time the X-ray diffraction pattern of crystalline DNA. This greatly influenced him to change the direction of his research toward the structural chemistry of nucleic acids and proteins. He soon found a job at the Cavendish Laboratory. It was here that he met Francis Crick and discovered their common interest in discovering the correct DNA structure. Together, Watson and Crick attempted to determine the chemical structure of living matter. When their initial research failed to produce results, the directors of the laboratory ordered them to end their investigation. But they continued their work in secret, and on February 28, 1953, they made a momentous discovery. The two scientists had determined the structure of the molecule deoxyribonucleic acid, DNA. Watson and Crick found that the DNA molecule is shaped like a double helix or gently twisted ladder. The two chains of the helix unzip like a zipper and produce their missing halves. In this way, each molecule of DNA is able to create two identical copies of itself. Using Erwin Chargaff's research that shows that A bases bind with T bases and C bases bind with G bases, combined with some crucially important X-ray crystallography work by Rosalind Franklin and Maurice Wilkins, contributed to Watson and Crick's discovery of the three-dimensional double helical model for the structure of DNA. Watson and Crick's discovery was also made possible by recent advances in model building, or the assembly of possible three-dimensional structures based upon known molecular distances and bond angles. Using cardboard cutouts representing the individual chemical components of the 4 bases and other nucleotide subunits, Watson and Crick shifted molecules around on their desktops as though putting together a puzzle. They were stumped when trying to understand how the different elements in thymine and guanine, specifically the carbon, nitrogen, hydrogen, and oxygen rings, were configured. Only upon the suggestion of American scientist Jerry Donahue did Watson decide to make new cardboard cutouts of the 2 bases, to see if perhaps a different anatomic configuration would make a difference, which it did. Not only did the complementary bases now fit together perfectly with each pair held together by hydrogen bonds, but the structure also reflected Chargaff's rule. Although scientists have made some minor changes to the Watson and Crick model since its discovery in 1953, the model's 4 major features remain the same, even today. Number 1. DNA is a double-stranded helix with the 2 strands connected by hydrogen bonds. A bases are always paired with T's, and C's are always paired with G's. Number 2. Most DNA double helices are right-handed. That is, if you were to hold your right hand out with your thumb pointed up and your fingers curled up around your thumb would represent the axis of the helix and your fingers would represent the sugar-phosphate backbone. Only one type of DNA, called Z-DNA, is left-handed. Number 3. The DNA double helix is anti-parallel, which means that the 5' end of one strand is paired with the 3' end of its complementary strand. Nucleotides are linked to each other by their phosphate groups, which bind the 3' end of one sugar to the 5' end of the next sugar. Number 4. Not only are the DNA base pairs connected by hydrogen bonding, but the outer edges of the nitrogen-containing bases are exposed and available for potential hydrogen bonding as well. These hydrogen bonds provide easy access to the DNA for other molecules, including the proteins that play vital roles in replication and expression of DNA. The initials DNA and the elegant model of the double helix became known around the world, as did Watson and Crick. Their discovery revolutionized the study of biology and genetics, making possible the amazing and revolutionary DNA techniques used by today's biotechnology industry. Since the DNA breakthrough at the Cavendish Laboratory, this understanding has started to change agriculture, with crops that contain pesticide, plants that make spider silk, and goats that can be milked for medications. This work has also led to a revolution in medicine, providing molecular insights into a variety of diseases from cancer to heart disease, assisting in speedier and more accurate diagnosis, and stimulating attempts to work with the genes to cure illness. It has shed new light on the origins of species, had vast impact on forensic science through DNA profiling, and even led to DNA computing. Thanks to the research done by Watson and Crick, we now understand a great deal about the genetic structure, and we continue to make great strides in understanding the human genome and the importance of DNA to life and health. We are just at the beginning. The biggest impact of the 1953 discovery has yet to be felt.